Dan Taratut, am I saying your name correctly? Terrific, is our first speaker. And he's going to be talking on mining the decomposition microbiome of spotted lanternfly, Licorma delicatula, I might be saying that wrong, for entomopathogens. What a cool talk. I don't know if we have many lanternflies here, but when I went to Hershey Park with my son, they were everywhere. And I was really, I mean, you're really torn because honestly, they're beautiful. But it's like, they're also like highly invasive species. And I'm sure Dan will tell us about all the ways that they may be problematic. I also was like excited to try to, not to be mean, but to try to kill them because I wanted to help. But I think you might have to kill them in like a specific way. That was a little overwhelming. Wow, Alan said that spotted lanternfly showing up in community garden. Okay, so they're really coming down. Oh, Adele said too that they're everywhere here. Wow, I guess I just am blocking out things that aren't mushrooms. Not too surprising. But anyway, so Dan is currently pursuing a master's degree at the Commonwealth University of Pennsylvania, and he is, I think, like I said, studying how fungal pathogens attack the spotted lanternfly and why fungal attacks on wildlife are rising worldwide. And this is a great connection. For those of you who were able to tune into our August meeting, about fungal epidemics, like this definitely is like a nice connection with that. So anyway, without any further babbling by me, thank you so much, Dan, for being here. We're so excited that we can have any small part of the work that you're doing and very excited to hear about it. Welcome.
Thank you so much for that introduction, Annie. I appreciate it. I'll go ahead and share my screen. Yeah, you know what? Aside from Basidiomycetes, I've always had a fascination for the other phylums that we know within our big kingdom of fungi. And every time I go out, I'm not necessarily even looking at the fruiting bodies of mushrooms. I'm kind of looking at what is colonized by fungi and kind of what insects are being parasitized and plants are being parasitized, because it really is just so deep in our environment. The mycelium fungi really affect it all. So without further ado, my name is Daniel. I am a second-year master's student at the Commonwealth University of Pennsylvania. I did my undergraduate work at Temple with Dr. Brent Sewell. My undergraduate work stemmed on spotted lanternfly ecology, actually, and then I also did a lot of mycology work with white nose syndrome in bats and that epizootic that has occurred for upwards of, we're coming on to about 20 years now. So I really kind of got exposed to invasion biology, mycology, and kind of general forest pathology in a lot of different work that I did. And so my advisor now was a close collaborator of my undergraduate professor. His name is Barry Overton, and him and I have developed this project called Mining the Decomposition Microbiome of Spotted Lanternfly for Entomopathogens. And so, for the outline of my talk, I'll kind of give a little bit of a history of spotted lanternfly and what kind of drives their invasion. And then I'll kind of change gears a little bit and really talk about what's being done to handle this invasion and kind of some of the novel tools that focus around fungi that are being kind of implemented now. So everyone sees my slide okay? I'll get a look for Annie for a thumbs up just in case everything's good. Awesome. All right. So spotted lanternflies came to Pennsylvania in Berks County, I believe it was 2014, on just one stone pallet shipment. And since then, they've really kind of gone about everywhere, it seems like. So even down in DC, it's quite a bummer. The county where I'm in, Clinton County in Pennsylvania, was actually just turned positive this past summer. So, we had kind of steered them away for quite a long time, but it's unfortunate how quickly they're spreading. And that's in part due to the fact that they have about 70 different host plants that they feed on. And some of those specialty crops include grape and hops. So for our wine drinkers and for our beer drinkers and enthusiasts, it's a bummer. It's definitely putting a little bit of a dent in that industry, and it's scaring a lot of those agricultural sectors as this invasive insect moves through. And again, this is really in part because there's a lot of different nymphal stages of this life cycle, and then the adult stage of the spotted lanternfly. The nymphs really feed on that soft vascular tissue of plants, and as the adults mature, they kind of start piercing bark and stressing out trees. And so you have nymphs feeding on certain plants when they're young, and then adults feeding on different plants when they grow. And so in general, you just get a lot of different plants stressed in our environment. And so you know, they're not necessarily killing off. You know, I will preface this. It's a good thing because spotted lanternflies aren't necessarily killing off any certain trees besides black walnut saplings and invasive tree of heaven, but they're adding to the additional stress that we see with global change. You know, as we have different precipitation patterns in the seasons, our forests are already stressed with so many different invasive plants and insects and animals. You know, this is just yet another stress for our forests. And so their prevalence has really been aided by the fact that this invasive tree of heaven has already been established for over 200 years in our country. So it's, you know, we had this insect come over in 2014, but their food source has already been rampant for so many years. And so, you know, this is definitely all things to consider. And so again, their dispersal is really propelled by their extremely fast maturation rate over the course of the summer, very high fecundity rates, their super generalist oviposition, that kind of 70 plus you know, a host tree and plants that they feed on, super inconspicuous nymphal stages. You know, they're so small. This little picture here of the first instars on the underside of a grape leaf. You know, they're very easy to hitchhike on a car, on a train, on any dispersal network. And so this graph here really kind of shows their predicted suitability. You know, you have Pennsylvania over here, and then you have this area in red where they're highly suitable to adapt and to thrive and spread out. And you can see that as far as California. Actually, spotted lanternfly have already been stopped on a wood pallet shipment in California on the border. So, you know, they're making their way around town, and it's quite sad. And so if you know, this is a really helpful diagram here for any botany lovers here, just kind of showing that when they're nymphs and they're younger, they're really kind of feeding on some of these fleshier, softer tissues of plants, some perennial flowers, vegetables, rose plants, grape, and as they mature, they kind of start feeding on some of these hard and softwood trees that are prevalent in our region. What's exciting is that since 2014, there have been documented cases of natural predation of spotted lanternfly, some egg mass parasitoid wasps, then you have different insects like spiders, praying mantises, you know, documented cases of natural you know, invasive pest control with our native predators. And so that's definitely very exciting, but yet we see this dispersal and we see this high prevalence of spotted lanternfly in new areas. So, you know, that's just to say that it's not enough, and that, you know, new management strategies are needed. I'm sure some of you have heard of the spotted lanternfly circle traps, or you know, circle or sticky traps that people put around the circumference of their trees. You know, this has helped to mitigate the spread, but it also traps a lot of non-targets. And so, you know, it's been really tough to kind of find a one shoe, you know, a one size fits all solution to really deal with this spread. And so something that's always been really interesting to me is the way that we deal with pesticides within agricultural systems, specifically with biological control agents, and even more specifically with microbial biocontrol agents. And so this kind of brings me into this slide here, really depicting that microbial entomopathogens, and those are essentially insect parasites, and specific biopesticides have really been underutilized in the States when compared to other countries. It's a market that may soon reach upwards of $8.5 billion worldwide. And so the production of fungal entomopathogens and bacterial entomopathogens has really been interesting to a lot of agricultural sectors. Specifically, entomopathogens that are able to successfully transgress the insect cuticle and actually infect the hemolymph, essentially the insect blood, of these cadavers germinate and essentially cause mortality. And this is a great diagram here showing Bavaria bassiana, infecting a beetle. And so, species candidates that are able to incorporate such penetrations have really been looked at in recent years for specific management options. With spotted lanternflies, there just aren't enough long-term solutions and cohesive management strategies. And so this is just really presenting a novel approach to utilizing fungi and avoiding kind of harsh synthetic chemical sprays that cause a lot of environmental pollution, kill a lot of non-target insects, and are just not safe for the environment or us. And so microbial entomopathogens are a natural path forward that I really believe the majority of the agricultural sector will start to employ in the States. A little bit of background for some common mycoinsecticides that are used. Maybe some of you have heard of Bavaria bassiana. It's a very readily commercially available, Bavaria in general is a very commercially available genera of mycoinsecticides, and there are over 55 globally used mycoinsecticides that use Bavaria as an active ingredient. Some use metarrhizum, some use Iceria. But in general, a lot of these cases, they're very host specific and actually show slightly low virulence. And so when it comes to spotted lanternflies, Eric Clifton at Cornell has actually utilized Bavaria and showed some good efficacy. But in general, it's kind of spotty in terms of the results of actually something that would work in a variety of different areas and something that would show high virulence against the specific insect. And so the question really arises, does the efficacy of commercially available fungi outcompete kind of native forgotten fungi that exist in the soil where spotted lanternfly are found? And so this really kind of brings me into my thesis in general and the overall objectives that I have been investigating. And I'll just read them off now. Really, I've been curious to isolate entomopathogenic fungi and bacteria from different soil types, actually using spotted lanternflies as bait to kind of show that, or test, I should say, the specificity and selectivity of different fungi to this kind of novel resource that has now existed in the environment since 2014. Subsequently, to sequence any isolates using DNA barcode regions specific to fungi and bacteria, that being ITS and 1 and 4, and then 16S rDNA barcode regions for bacteria. Later, to determine the suitability of these species as entomopathogens in various application settings, based on an extensive literature review I've been conducting. And then lastly, which I think is the most exciting part of my work, to select best species candidates for actually incorporating efficacy studies in my thesis on egg masses and first instars. And that is more so done to essentially provide a solution to really target these kind of first life stages and stop them. They're an annual insect. They lay their eggs and all the adults die. And so in my brain, from an invasive species management perspective, when you target something earlier on in its life cycle, you're reducing its population a lot quicker than if you're waiting until it's an adult and reproduces. Unfortunately, a lot of avenues of research just haven't really thought that through, I suppose. And so, tackling my first thesis objective, I employed a Gallery bait method again. First used in Zimmerman et al. in all the way back in 1986. And I used 5 obscure soil types ranging from Pennsylvania mine, tunnel, railroad, compost, weird, underutilized and understudied soil types for native fungi, in addition to 2 sterile controls. And I basically baited these cadavers on these soil types. I used 10 cadavers per soil sample. And I didn't surface sterilize them. I really wanted to see what the kind of microbiome of spotted lanternfly entailed. I used an even mix of males and females, these females kind of with their big yellow bellies towards the end of the season when they're full of eggs, and some of these smaller males right here. So a mix of males and females were used, and they were frozen before I had initiated this part of the experiment. Again, they weren't surface sterilized, and they were left out for 30 days at room temperature around 20 degrees Celsius. And so from this kind of came some interesting results. You know, we really kind of wanted any fungi, any fungal spores that were within those soil types to penetrate the hemolymph of these cadavers. And so we really focused on the internal microbiome of these insects. And so we actually followed an old school protocol from an old paper that looked at fungal pathogens in corn plants all the way back in the 60s. And we used a surface sterilization technique to target these species that were capable of this successful cuticle penetration to the spotted lanternfly hemolymph. And so this trial yielded the successful isolation of 4 different morphotypes, which we later sequenced with ITS analysis. And I just think these pictures are so interesting, you know, all these microfungi that are colonizing these spotted lanternfly cadavers. Most of them were mucorales. You can see some of these mucor-filamentous fungi kind of colonizing the legs and abdomen, but we did get some really cool, some other cool colonies as well. And so to actually go through the isolation methods for these entomopathogens, we used a medium called DOC2 PDA. It's a selective medium known in the literature to target many entomopathogenic species. And we specifically use this formulation.
How'd it go? How'd it go?
Sorry about that. I'll just keep going.
Sorry, if you guys could mute yourself, if everyone could mute themselves except for Dan, that would be great.
Okay, go ahead.
But at any point in this talk, I do talk super fast, but please interject.
Good,
if
fine,
you
okay.
want me to go back or anything.
Chris Bowen, could you just mute yourself?
Lateral front.
And front.
Or maybe Tom might be able to mute Chris.
I'm looking.
I can't.
Lateral front and step. Yeah. Okay.
Where is he? Ah, there he is.
Okay. So sorry, Dan.
No worries. It keeps
That's
Okay.
one of the problems with having a big attendance. It's hard to find somebody who's talking.
No worries. But getting back to kind of some of our techniques with really fungal isolation of these entomopathogens, we use the specific medium that is known for entomopathogenic species, really based on its performance of kind of high growth rates for a lot of these entomopathogenic species, but also with the addition of a component called crystal violet within this medium. It's used to reduce any saprophytic fungal outgrowth, as well as bacterial growth. And so this was a really interesting medium for me to use. It was kind of the first time I really went into media development, and you know, it proved very successful. You know, we externally sterilized those cadavers from the previous few slides, kind of ground them up using sterile technique, dilute plated those you know, there's those solutions into a 1 to 1,000 dilution, and then subsequently morphotyped and separated colonies, and really kept track of colony forming units for later Shannon-Weaver diversity calculations, you know, to couple that with any sequence data that we had received later on. This was kind of just to give us a prevalence of the concentration and quantity of different entomopathogenic species that we were observing. And so some of these plates here are just showing our different soil types, and from left to right, you know, the 10 to the 1 to 10 to the 3rd dilution. And so overall, I will say, you know, this is kind of what some of the plates look like after 10 days. Those mucor fungi that you had seen colonizing some of those cadavers really took over our plates. But from a few of our sites, we were able to isolate some single colonies and kind of go through with our analysis. And we actually determined that our Fusarium species, specifically Fusarium solani and Fusarium fugicori, were the most common entomopathogens that we had isolated from these spotted lanternflies. We had hypothesized that this bioamplification that occurred from our soil or occurred from the soil and not necessarily from the cadavers, just because of its high abundance in most soil types, and not necessarily being a selective pathogen to insects within the order that spotted lanternfly fall into. And so we had excluded that from DNA sequencing, perhaps from some external contamination from the sterilization that we had done. And so based on that, in addition, this whole project actually really started when my advisor from Temple, my undergraduate advisor, sent me some spotted lanternfly eggs that he was collecting for some of his ecology work. And they were colonized eggs, full of mycelial outgrowth. And, once I had gotten to Lock Haven here and started my master's in the first few weeks, I realized this is pretty cool. Like, over consecutive years, we're seeing that these broken up egg masses kept in sterile jars, collected using the utmost sterile procedures, are just naturally colonized by something. And you can really see that here on this image on the right, because you have this egg mass here, essentially this portion right here, completely colonized by mycelium. And this is aided with a calcifluor white stain. It's a very common fungal fluorescent stain that picks up on mycelial fragments and spores. And so you can really just see the intense colonization that goes on just with one egg. Based on just this observation, we not only saw Fusarium again isolated, but we also saw Cladosporium. And that's all these kind of little lemon drop spore shapes here, in addition to the elongated, very kind of morphologically easily identified Fusarium canidia too. So, it was really interesting to see this again and kind of see this colonization in a different aspect of my work. And so, based on just everything that, all the different avenues of isolating fungi from egg mass material, from adults, from the baiting experiments that I had done with my advisor, we had just gotten a good amount of entomopathogens that were really well known in the literature, but very forgotten in commercial entomopathogenic development, or entomopathogen development, I should say. And so we had some penicillium, a few Fusariums, Cladosporium, Aspergillus, Iceria, and you can just see these beautiful canidiophores and microscopic images here of just some of these that we were able to isolate. And so moving on a little bit, we not only wanted to look at fungi, but we also wanted to look at bacteria as well. There's unfortunately no one size kind of fits all selective medium in the literature for isolating entomopathogenic bacteria. And most give very variable results. So we actually attempted to create our own, which was really fascinating and cool to do, especially as kind of a first-year mycology student here at Lock Haven. But to develop that, we actually wanted to incorporate the spotted lanternfly itself as an ingredient. And so we did some X-ray fluorescence data, essentially kind of better understanding the elemental composition of not only a completely mature adult cadaver, but also the egg mass material, including the eggs. We had some unhatched eggs that we ran under this XRF machine and found that the elemental composition was quite similar. And so further examining these similarities and differences, we discovered some interesting results. We actually noted that there was a big pH change when we were autoclaving our our media and noted that between egg masses and adult cadavers, the egg masses turned our medium very acidic. And for medium formulation, we didn't want a very acidic media that would inhibit bacterial outgrowth. And so again, why is that pH change important when you're developing, any type of selective media, especially targeted towards bacteria? It would just reduce its outgrowth. And perhaps this is in part due to some kind of organic biomolecules that are a component of the egg masses that reduce pH. And so overall, in our medium formulation, we had used the adult cadaver instead. That had a pH of 6, whether we autoclaved or not. And that was kind of our active ingredient, in addition to a half-strength tryptic soy agar formulation. And so this was really interesting. We kind of went through a similar procedure, but dilute plated up to 10 to the 8th, which is a much higher dilution. Bacterial colonies are just so freaking frequent and pervasive. Bacteria are just way more numerous than any fungal spores may be. And so we got way more bacteria than we did fungi, about 18 successfully sequenced samples comprising of 13 different species. Sphingiobacterium and Pseudomonas were the most common. Again, we followed a very similar internal microbiome isolation protocol that we used for the fungi, just to really target any bacteria this time that could be penetrating the spotted lanternfly cadaver. And so, it's really unclear whether some of these bacteria, just because some of them are really environmentally common bacteria, whether they came from the cadaver or were already present in the soil that we had actually baited on. And so, we were kind of only looking at the internal microbiome and really kind of focusing on the things that really did come out after this kind of surface sterilization protocol that we had done. And so, all of that considered, moving on to kind of my fourth component of my work, which is to test the efficacy of some of these entomopathogens directly on the spotted lanternfly. Our goal was really to distinguish which of these would be the best candidates for this study. And so, kind of as I mentioned earlier, targeting the early life stages is really the best to reduce populations in subsequent years. Overall, based on the literature, spotted lanternfly are in the Hemiptera order, and insects within this order are actually, they die a lot quicker when they're exposed to different fungi than they are to bacteria. Fungi are just much more effective than bacteria at killing insects with the piercing and sucking mouthparts that the spotted lanternfly use to feed on so many different plants. And so out of all of the different species, both bacterial and fungal, that we had isolated, we had kind of come up with a comprehensive list of Fusarium and Cladosporium would be our main fungal agents, and then Pseudomonas would be our bacterial agent that we would test. All of which are very forgotten, opportunistic insect pathogens, both bacterial and fungal, and all comprise of a very wide host range and are just very common in our environment. And so here's a beautiful tree of different Fusarium complexes, about 10 Fusarium complexes that are pathogenic to a variety of orders of insects. And so I just thought this was a really cool chart here, just highlighting Fusarium itself and how pathogenic it can really be to a lot of different insects. And not only, we also kind of wanted to incorporate the actual variation of egg masses. I'm not sure if spotted lanternflies are just starting to come out in DC. Maybe a few of you will know that it's the adult season and they are laying eggs. If you look under sugar maples or oaks or tree of heaven or even grapevines, you'll see these types of egg masses, and this is exactly what they look like, but they do have a lot of natural variation. Sometimes the female adults don't necessarily cover their eggs in this kind of protective covering, And sometimes they're just partially covered, fully covered, or a little bit weathered if it's, let's say, a very windy or rainy day. And so again, this egg mass covering is very variable. And there's a chance that this protective casing can harbor fungal spores and subsequently reduce first instar hatch rate success. So this is kind of another thing that another variable that we were examining in these efficacy studies. And so I promise I'm getting to the end here. I know I talk a lot, but this is just a quick flow chart, maybe to just visually show what we really did with some of our entomopathogenic candidates. After growing them to pure culture, we had developed spore suspensions between 10 to the 6th and 10 to the 8th concentrations. This is kind of the baseline concentration that's used in a lot of commercial grade formulations of entomopathogens. From there, we had actually piggybacked off of some protocols from Cornell that were actually just spraying these spore suspensions directly on nymphs with an airbrush, but instead we had sprayed them on egg masses. And so we had a treatment group, obviously the kind of spore suspension or bacterial suspension group. We had a negative control group that had absolutely nothing sprayed on it, and a positive sham control that just consisted of sterile water. And so all of these different egg masses were sprayed with all of these different treatments. They were staggered and incubated at 20 degrees Celsius for 22 days. Usually nymphs, when fully mature, eggs when fully kind of through their egg mass maturation process, would hatch within 2 weeks. So we gave them a little bit more time, and kind of mimicked environmental humidity around May, which is when they're usually hatching. And so from there, whether the nymphs hatched or they didn't, we evaluated each treatment type. We kind of snuck a piece of egg mass material, whether it be an unhatched egg or a hatched egg, and exposed it to that calcifluor white stain, that kind of image that I showed earlier on with that fluorescence, to confirm any egg mass colonization that may have or may not have occurred. And in addition to that, we also documented hatch rate success. And we did that by actually literally counting whether these eggs were hatched or not. And so, this is just kind of one of the trials that we had put in our experimental growth chamber, in addition to our 8 different treatments that we had deployed. And so our Pseudomonas fluorescens was our bacterial candidate. And then we had all these very common, but again, underutilized fungal species that we used as treatments in this study. And so you might ask why there's 2 egg masses per plate. This was just to account for any internal variation. There's always variation in terms of how things might mature or develop, whether they're looking up at the sun or looking down at the ground, exposed to UV light and factors such as that. So even in this picture here, you can see that from just an example egg mass, some nymphs hatched, and you can kind of see that with this like open flap here that's popped up. And then some just never opened. And so those nymphs just never fully matured all the way. And so to kind of account for some variables, all of the egg masses that I had used in this study were collected from one site on the same day, and totaled 4,337 eggs total that we had counted. So quite a lot of spotted lanternflies that we could have released, but chose to do some good science on. And so not only that, we had inoculated all these egg masses with their specific treatments. After observing colonization of fungi in these eggs, we had actually wanted to kind of fulfill Koch's postulates and re-isolate what we had initially sprayed on these eggs back out, just to know that what we were seeing under the microscope was really what we had actually meant to be there. And thankfully, everything that we had, we fulfilled Koch's postulates for absolutely every treatment, obviously aside from the sham control. everything that we had inoculated, we got to grow back out. And what's interesting is that not only did we re-isolate every original pathogen that we had kind of treated for, but we grew out Cladosporium, which is a very known entomopathogen and a common fungus in our environment, back out each and every single time. So we're starting to hypothesize that perhaps there are some native fungi that are kind of reducing hatch rate success and naturally colonizing these eggs. And so the next 2 slides, I'm actually going to show you guys some really cool images of how we visualize this fungal colonization. And I'll tell you a little bit about how we're kind of taking this data forward. These images here are that calcifluor white stain. But this picture on the right-hand side in the upper right is just an original 40 times magnification image of an egg mass material in a random field of view. Once we apply the stain, you can see in this exact same field of view, you have this mycelial fragment, these mycelial fragments completely colonizing the egg mass. And so, using our computers here, we had overlaid these images, and you can kind of see this type of colonization that goes on such a microscopic scale within these egg masses. And so here's another example of much more bioamplification of these fungi. And I could look at these pictures forever. I just think they're beautiful. But it really it's a proof of concept that these spotted lanternfly egg masses are being naturally colonized. And not only that, but these different treatments that we sprayed with may or may not be reducing hatch rate success. And so utilizing the data that of whether nymphs hatched or they didn't, and actually correlating it to fungal colonization within these eggs, with some statistical analyses, we can really show which agents, which candidates are really causing the most mortality of these eggs. So again, it's really just a novel way of using fungi as a biocontrol tool, and kind of and there's a big application of these microbiocontrol tools in a variety of different areas. But of course, before I kind of wrap up, there's an importance to using biocontrols, a big ecological importance. Many times in our history, we've released things into the environment and things have gone south because there's a lot of different safety effects, side effects, safety considerations and host range transgressions to really keep in mind. And specifically with mycopesticides, whether you're spraying them in a natural, cultivated, or even an urban like city landscape, whether it's a plant pathogen of a specific crop that you might be spraying on or near, the timing of application in the season, and then of course the concentration of application. And so, just to really kind of drive things home here, a lot of this data is in process, a lot of this data processing is in progress, really kind of utilizing different computer software programs to quantify the amount of colonization within some of those images, and correlating that to hatch rate success is kind of the last and final part of my thesis. But it just really brings home a point of mitigating the spread and damage caused by these spotted lanternflies, and kind of using a novel technique. And so again, reducing the first instar and the egg mass really phase is kind of the best management option within the agricultural sector to protect a lot of economically valuable specialty crops such as hops and grapevine. As many, with a lot of commercially available strains, they hold a lot of sensitivity to abiotic stressors and naturally show low virulence. And so it's something that I feel passionate about, that other entomopathogenic tools really must be explored, especially for the use as microbial biological control agents with this pervasive insect pest. And so, we're kind of we're getting somewhere, and I think that these results are quite novel and quite fascinating. And so, I'd just like to acknowledge a few people that have helped me. My collaborators at Temple, my committee members, and of course, everyone here, the Mycological Association of Washington, D.C. Thank you to all of you for this opportunity to, for additional funding to do DNA sequencing and to purchase lab materials. It really means so much to do this type of work, something I'm so passionate about, and have the support of a wonderful association such as yourself. So thank you so much. Just some references of the talk I did. And hey, I'd love to answer any questions that anyone has. I know I talked for so long. I'm sorry if I'm over time.
No, that was great. Actually, Dan, that was terrific. And it was really an excellent talk. And it's just really awesome to see. I mean, it's like really cool that to hear about something that is literally happening right now. Like we talk about all of these changes to our environment, but I mean, it's sad, but we're watching this happen in front of us, and it's like not debatable that it's happening. It's not like, oh, things changed by half a degree. Like we know it's real, but a lot of people don't think it is. Like you can't miss the lanternflies. They're definitely there. So I think this was really awesome. You definitely have a bunch of questions in the chat. So some Adele was asking how do you apply the fungi to treat an area? So like let's say you got the go ahead, how would you do it?
Sure. You know what? There's a lot of different ways that you can apply something. At the end of the day, it could be as easy as wrapping some of these egg masses, obviously a very mechanical approach to just removing them. But, in an area, let's say, such as, I've been seeing videos of New York and some of these really high metropolitan areas that are just infested with spotted lanternfly egg masses, and even, all the way up to adults. But funny enough, there's no vegetative growth to be supporting them, in these areas. And so, again, it really depends on what type of mycobiocontrol you are spraying. I see a few comments here that fusarium is nasty. Fusarium is a known plant pathogen. Some species complexes of fusarium I would not use around, vineyards because they are known pathogens to grape. But let's say in a natural forest system, let's say a natural forest, cladosporium and Pseudomonas and fusarium, they all exist naturally. And so really targeting where, kind of going into spotted lanternfly ecology and behavior and understanding where they lay their egg masses would kind of be the first point for me in terms of actually developing management strategies to spraying some of these mycopesticides. But it's a loaded question. It really, again, depends on what you're using, how you're using it, and where. So there's a lot of different ways I suppose I could answer that.
No, that's great. Well, Mitch was asking, you mentioned the low pH in eggs. Do you think that's like a natural method to reduce the susceptibility of being colonized? Or?
Sure. Yeah. You know what? I'm working with the chemistry department here at Commonwealth to investigate that a little bit more. I think there is some sorts of biomolecules that are in this kind of very hydrophobic egg mass covering that definitely could hint to a higher acidity. There's also a little bit of elevated copper in the egg masses than there are in the adults, interestingly enough. And so, that perhaps might play a role in the pH. Definitely a question I'm still investigating and hope to have a more definitive answer to.
Actually, I have a question that is might be chemically related, although I'm not sure, but we were just sort of talking in the chat about how both fusarium and Pseudomonas both can cause like onychomycosis or nail infections. And then, excuse me, also I think fusarium has been spread via contact lenses. And so I guess I was wondering, is the ability to penetrate nails or contact lenses related to the fact that it can get in the exoskeleton, and do we understand what chemicals are allowing that to happen? Or
Sure. That's a great point. I mean, there's just so many secondary enzymes that are these secondary degradation enzymes that are very variable depending on what species you're looking at, what genus you're looking at, and some are just more some are stronger than others. Some entomopathogens just aren't as virulent as others. I really would attribute that to the specific secondary degradation enzymes that perhaps one entomopathogen might have over another. And especially when that comes to us, I'm sure if anyone's seen The Last of Us here, like fungi are coming for us, right? And so these enzymatic pressures and these kind of evolutionary and adaptive mechanisms, I'm sure that certain fungi have that others are just maybe slightly catching up to. But that's definitely a great point. Again, some things I wouldn't use in certain environments and some things I would.
Annie, I had one question as
Great.
well. So you highlighted 2 isolates, I guess, that you'd done the ITF sequencing on.
Yes,
Do you guys have plans to do further characterization of those?
we would love to. We would love to sequence them a bit further and really kind of and do some further characterization, definitely. With cladosporium kind of being our Fusarium and cladosporium are really our main targets at this point. And Fusarium, we really only isolated from those baiting experiments. Whereas cladosporium, we're really seeing often in multiple life stages of the spotted lanternfly. And we're really just seeing them naturally colonizing basically every single egg mass that we had isolated from. So our focus is actually shifting a little bit. We're definitely interested in specific species complexes of Fusarium that aren't plant pathogens or aren't as aggressive plant pathogens. But we're really kind of interested in this characterization of the cladosporium that we've been working with and seeing a lot in our isolations.
Okay.
Yeah, so I mean, I'm only in my second year, and I still have about a semester and a half. I finished this program in May, so plenty of time to answer some of these kind of unanswered questions, and hopefully I can come back with a good conclusion for you guys.
Yeah, we definitely would love to hear from you again. And do you know, this is probably like a loaded question, but do you know, are you going to want to continue with like the school route, or are you planning to go work in the field, or what's your plan?
Great question. Hey, it's hard. I don't know what I'm eating for breakfast tomorrow.
Horrible question, like literally horrible question that everyone hates being asked, but old people love to ask. I think I like officially have become old because I'm like, what's next for you?
I love it. No, I mean, mycology has always been a deep passion of mine, and I think the kind of the ways in which it intersects with invasive species ecology and kind of the microfungal route of really investigating this kind of unknown world that we're next to is definitely something that's super interesting to me. So I think the school route is maybe perhaps a PhD ahead for sure.